Live first.
Okay.
Good afternoon,
Good
everyone. Uh,
afternoon. Yeah, please go ahead. Yeah.
good afternoon, everyone. Request our principal who's just joined us for a short time, Dr. Ashok Wadia, to say a few words about Dr. Sagarika Damle, our speaker for today.
Good afternoon to all of you. I'm glad that under this COVID situation and the lockdown, when the colleges are not open, everything is happening virtually. The botany department of Jain College is organizing various activities, and one of that is today's lecture. It's a series of lectures which are conducted, and this is a very important topic because everything is digital now. And in the biology, one of the important role which is played is by bioinformatics. Huge data, so the big data which is now is getting control, and analysis of the species, genus level, all those things which, if you want to compare, that can be done. So proteomics, genomics, all those things come. So I'm very glad to see that they are able to get the speaker like Damle madam, um, who is expert in this particular field, to address this issue. Last time you were lucky enough to have the person who had given you the insight of HTPLC and how to use it, all those kind of things. So today is another such session, and students, please take advantage, ask the questions, get insight of it, understand the things, because that is your future. Thank you very much for inviting me on this particular platform, and thank you, madam, for accepting our invitation, and entire team. Unfortunately, like every time entirely there for the entire session, but I'm already on the panel, so will be, actually I'll just switch off my mic over there and my video, and I'm talking to you. So thanks again, and continue with this particular session. I will take the feedback later on from everyone. Thank you.
Thank you so much, sir.
Thank you, sir, for those encouraging words. It definitely means a lot to us. Good evening, everyone. We welcome you to the series of lectures conducted by the Department of Botany, Jai Hind College. Today, our speaker, and it gives me immense pleasure to introduce this beautiful lady to you, Dr. Sagarika Damle, an associate professor and head Department of Life Sciences at KC College since 2014. She's been in the field of education for a very long time, and I know her for more than 30 plus years. Ma'am has been at the Institute of Science, and that's how we came across each other. She's a research guide for PhD and MSc degree in life sciences for botany as well as environmental sciences. Ma'am has 17 publications to her credit, plus she's edited a book with the title, she's had a chapter in a book called Agriculture, in a book called Science and Technology in Ancient India. Since the talk today is on bioinformatics, I would certainly like to mention that she has 13 gene sequences that have been submitted at NCBI's GenBank from 2017 to 2019. And I think very few people would have that to their credit. She's on a number of committees, a lot of academic positions. She's a board of studies member. She's chairman board of studies for life sciences from many, many years that I hear. She has taken a number of research projects, but she's also working on as a research associate on environmental impact assessment projects by the central and state government. So besides being a environmental person, she's taken keen interest in bioinformatics over a long period. She is somebody who has been promoting research at KC College for a very, very long time. And that's why she's been the convener of undergraduate research program Jigyasa, which is held at KC College every year. She's the coordinator of the STAR College status. What I'd like to mention is that such a beautiful personality, Beauty with Brains, has got a very simple motto in life. She says, meeting any challenge with a positive attitude. And you can see that positive attitude in her every time you meet her. Ma'am, it's an honor to have you here today. Uh, Please begin with the talk. I will not keep you waiting and the audience waiting for this particular beautiful lecture. Over to Sagarika ma'am.
Thank you, Archana madam. Thank you, Devangi ma'am, Bharti ma'am, and my dear friend Sangeeta ma'am. And of course, thanks to your principal, who, in spite of his busy schedule, he was with us for some time. And I'm really glad. I love to be with students and teachers. And when teachers are my friend, I think that's the best thing that can happen. Regarding bioinformatics, I think though I actually had sent her my CV and I said, don't say everything that is there written because that's the normal thing that we send across. But I came into bioinformatics as an accident. And I can say this now after so many years that because all the senior teachers, they said we are not, going to take bioinformatics, which was just introduced as a new discipline that time for MSc, and I had to do it. I didn't know nothing about bioinformatics. So if I can come this way and I can learn few things, I think you people are very, very, very fortunate that you are so tech savvy, you are so equipped with other knowledge that last 30 years have brought into science. I think it's a beautiful journey that you will enjoy if you want to get into bioinformatics. And as I always believe, you need to have passion about anything and everything you do. Even if you do not like it, be passionate about it. Later on, you just might like it. It's an accident. So I'm going to share my screen, thanking once again to Devangi ma'am for this opportunity and all the other teachers. Can I start my presentation, please?
Please, ma'am.
Thank you. So can you see my presentation?
Ma'am, it's not
Not
visible
yet,
yet.
ma'am.
Can you see it?
Not yet.
Not yet, ma'am.
Not yet. Okay. Let me again try once again. Share.
Yes, ma'am. Now I can see it.
You can see it?
Yes, ma'am.
Yes, ma'am.
Okay. Can you see the full screen, please?
Yes, ma'am. It is seen bioinformatics.
Full screen, right?
Yes, ma'am.
Oh, thank you. Okay, great. So my talk today is on frontiers of present and future modern science. And I'm sure that we all biologists need to be really, really proud. And why I'm sure about this, because when the word bioinformatics comes to your mind, the first word is bio and the second one is informatics. So, I'm always more biased towards biologist taking up the career or taking up the challenge of being a bioinformatician. So therefore, what is bioinformatics? I'm sure each one of us know it's an amalgamation, a beautiful synergistic platform that has come into existence because two very powerful fields have merged. One is biology and one is informatics. So the education of biologists in the use of advanced computing tools is what bioinformatics is all about. And let me tell you very honestly, I am not a computer scientist, nor am very well versed with writing programs. But then, do I have an upper hand as a biologist in bioinformatics? That is because I can analyze the sequences that come to me. I can look at the DNA, RNA, protein, and I can make sense of what it might be doing. I know from the minutest molecule to the largest tissues or systems what they are doing as a biologist, and therefore in the field of bioinformatics I have an upper edge or upper hand. So I always like to tell stories to my students, and I think everybody likes to hear a story. So I'm sure people must have heard this story about four blind men and an elephant, and each blind man tried to feel a part of the elephant, and then he tried to imagine what this structure that I am looking at or I am feeling. And something similar happens to bioinformatics, because bioinformatics is an amalgamation of many subjects, many disciplines, and each discipline feels that, oh, it's mine. And that is the success of bioinformatics. So it is biological sciences, may it be statistics or biostatistics, may it be chemistry because the structures of molecules, may it be biochemistry, how they interact with each other and how the molecular processes are driven, biochemical processes are driven, and of course, the informatician, we cannot forget him, the computer scientist or computer analyst who says, oh, come on, I'm the boss here and that's mine, you know. But that is the whole beauty of it. This is the 20th century model of science, and it is called as bioinformatics. It is an interdisciplinary approach to solving queries of biology. So if I try to break it into two parts, so first is biology, and what do biologists do in bioinformatics? Collect molecular data, DNA, protein sequences, gene expression, etc., etc. Remember, there is an avalanche of information, avalanche of things coming in every day because of the advent of all these sophisticated machinery that we have at our disposal. So sequencing a genome is not much of a problem, and it's not so costly as well. I'm sure some of you have heard about commercial enterprises like 23andMe and Map My Genome, where they will take your saliva and sputum sample, and they will just tell you your DNA markers, and they will tell you what disposition you belong to, what kind of diseases you may have in future, or what kind of race of ethnic group do you belong to. So all that can be done at a switch of a button nowadays. Whereas informatics or informatician is the computer science and the information uh, analyzing part through machine device. And of course, finally making sense of it in the true biological sense is the work of a biologist. So therefore, computer scientist, he could be a mathematician, he could be a statistician, and he would develop tools or software programs or algorithms to not only store the data, we can add to the data, we can edit the data, we can retrieve it, and we can analyze it. And very, very important process, or I would say the most important part of the entire bioinformatics gamut is that most of it is free. Aren't we happy? I mean, when the whole human genome controversy came up, I'm sure you all know, and now you must, by now, all those students who are listening to me, they must have understood that I'm really jumping from one discipline to the other. I'm talking about chemistry, I'm talking about biotechnology, I'm talking about human genome. So please remember, in your mind, there should be no walls right now, that I belong to biology, I belong to botany, zoology, life sciences, biotechnology. Please don't. That will restrict your expanse, or it will restrict you to get foraging into this vast field of bioinformatics. Okay. So though I am a plant biologist, okay, when I come to bioinformatics, I think about DNA, RNA, and proteins. Like, so ये जो अर्जुन और वो fish की आँख थी ना, uske jaisa, whatever I am aiming at, that time I'm going to think only that. So it could be a plant genome, it could be an animal genome, but the beauty of it is, because I'm a biologist, I may know what is different between a plant and animal genome, right? So bioinformatics is the study to give answers to the biological questions. But remember, this all data is mostly in the molecular form. So why do we want to do bioinformatics? Some people, when it just came in, people said, oh, these are all fancy things, the old wine in the new bottle. Why are you calling it as bioinformatics? Because remember, as I just said, there is an avalanche of data, vast biological data, which has been created since the advent or since the finishing draft of Human Genome Project. Many of us thought that, oh yes, Human Genome Project decode ho gaya, that means we have got answers to all our questions. No, actually, many of the questions then arose, due to which now the further research is happening. So finishing human genome or decoding the human genome, that means knowing where each gene is on which chromosome, was just the beginning. Okay, so the bioinformatics platform allows us to collect this data, or obtain this data over www. I'm sure your teacher must have talked about it. You can organize the data, you can store the data, you can communicate the data, and you can manipulate the data. Of course, when I say manipulation, I'm talking about the biological manipulation, and I'm not talking about the unethical part of data manipulation as such. Of course, accurate and quick analysis of the data, it saves time definitely. Imagine if you have to write 30 amino acids in a polypeptide chain, on a piece of paper, and then, you need to convert it into a DNA sequence corresponding to its codons. How much time it would take and how much space on the paper. Whereas if you do bioinformatics, and I'm sure ma'am must have shown you gene bank up till now, you must have seen some protein databases, and you must see the humongous amount of information available. And that where the problem lies. Many of us, including us teachers, we come into this field and we want our students also to, get acquainted with this field. But what we do not realize is that this is very Greek and Latin to most of our students. So whenever we look at these terminologies or these databases or all these software tools, we need to start from scratch. And I'm sure many of the teachers must have experienced this. You finish a database and then students ask you like Ram ki Sita kaun thi? And then you have to go back to the database and say, oh, I think I need to explain what database is. So please remember, let's not be in a hurry. I may say today in my talk that, the best promising field in bioinformatics is drug designing. But to reach to that stage of drug designing, you need to know what data means. What is a data, biological data, molecular data, how DNA looks. Okay, you need to know most basic things, and then only you can go to docking and you can go to drug development. Things don't come easily, and please, those who wish to get into bioinformatics, it's a fantastic field, but a lot of hard work, a lot of new language learning things. Do not get demotivated by amount of data that is available on the net. You go there and you will find like lakhs and lakhs of entries for anything that you are searching for, and then you don't know which data to look forward to. So one of the reason why I feel students should take a bioinformatics is, suppose you are facing some problem that this particular syllabus, if I try to search on the net, I'm not really getting what I want and I'm getting so many other things, right? So for you, it would be very nice if you curate all those databases that you see, or you curate that information, and you make a kind of a list of resources that can be used for your syllabus. So that's a great start somewhere. That if I'm studying proteins, if I'm studying DNA, so what are the sites that would give me the best way that I can look at my data, the way it can be related to my syllabus? Because if you try to chew too much in one go at the early stage, then you choke. Okay, I'm sure you want to take it easy. And as I was talking about Human Genome Project, that is when all this started. So what kind of biological data are we looking at? We are looking at, and I'm going to come back to this slide later on. But so सबसे पहले, what is encoded by our genome? Definitely we want to know that. And do you think that all our genome sequences, or all our genes, we know what do they do exactly? I'm sorry, but no, we still do not know, because they are acted upon continuously by environmental pressures. And what really happens is then epigenetics. And I'm sure somebody else is going to come in for you for epigenetics, because that's another a fantastic, I would say, a very, very interesting field to get into. How is the genome information expressed? In terms of what? I mean, in terms of genes or in terms of proteins, right? And then the proteins make certain reactions go here and there, and then they form their products, and then all the metabolic processes are driven. How does the genome differ from others? This is another very important question. Our search for ancestry, our search for which was that particular first cell from where we all have come into existence. So how do they differ from each other? And how does the genome interact with its environment? As I was just saying, no two genes will behave in the exactly same manner in two different species if their environment is different. And all this we have been learning. So when you get into the field of bioinformatics, do not forget what you have learned before. Do not टेक फॉर ग्रांटेड कि ये सिलेबस मैंने ये सेमेस्टर में खत्म किया, उसको अभी मुझे नहीं देखना. Okay, so this is very simple. I'm not getting into the nitty-gritty of cellular view, but I'm going to tell you, if you are looking at bioinformatics, you should know transcription very, very well. You should know what is splicing. That means how do pre-mRNA get into mature mRNA, because the mRNA is the one who's going to make the proteins. Then how the mRNA is transported. In fact, I would say entire gene regulation part of it is something as a biologist you must know, because finally we are talking about proteins, of course, and finally we are talking about what proteins are doing, what is their function, how are they getting into metabolomics, and how are they then finally giving rise to a phenotype, or whatever maybe the expression that it is looking at. But before that, the regulation part of the gene is extremely important. So if you do not have these basics, then bioinformatics becomes a little difficult. Now, as I said, it is the old wine in the new bottle, and the terminologies have changed. But just look at this chart and you will try to understand this. We talked for so many years about the biological series. Then we came into the omics series, like genomics, proteomics, metabolomics, and pharmacogenomics, a fantastic field again. And I'm using this word again and again because I'm really stricken by all these things that happen in bioinformatics, pharmacogenomics for that matter. And I would like to tell you something interesting here, if anybody knows what pharmacogenomics is, and I'm so missing my usual class where I can ask students a question who are sitting in front of me, but here I have to give all the answers. So the burden is pretty high on my system here. But pharmacogenomics, I'm sure you know that it is something to do with pharma and something to do with genomics. So we are actually looking at genome-based pharmacology products. Okay, and finally in informatics series, it is going to get into the area of health informatics. So as we see biological series as a hypothesis-driven biological problem solving area, we see informatics series as data-driven computational technique. And as I want to move on from here, before that, I'm going to just give you a very quick, interesting fact from our yesteryears, that a very good physician, a doctor, which were, of course, Ayurvedic practices before actually allopathy came into our lives, okay? That good a doctor, a Ved, he used to just feel the pulse of a patient, and he would come to know what kind of system, the Tridhatu system, that the patient had, and what was not right, which of the three dhatus were not functioning properly, and that just by feeling the pulse. Imagine that. And he could then prescribe medicine A, I'm saying A as a particular medicine, to a particular person with particular type of constitution, body constitution. But he would not prescribe the same medicine for the same element to another person. He would prescribe medicine B because he knew his body constituency is not what first person's constituency is. What was that all about? Nothing but pharmacogenomics. We had all that knowledge before. Now, I'm so sorry to say, but many of the doctors have to tell you, go through all these tests, which all are machine driven. And then when you come with papers, then they will look at it and they will tell you what is wrong with you. And unfortunately, they will prescribe the same medicine to 10 people and only 2 of them will get better. So I think pharmacogenomics is one such magic wine. I would say it's a magic wand, which will tell us if your SNPs are showing a particular type of allergy or particular type of reaction to a particular drug, then that person should not be given that drug, especially in case of cancer treatment, in case of all those high-end drugs that are prescribed for rare diseases. Pharmacogenomics plays a very important role. So this is a new era, new terms, and therefore, as I said, we need to know these terms such as computational biology, computational biomodeling, mathematical biology, and network biology. Now, simply here, if I was a biological student, the word mathematic itself would make me run, 10 meters away from whatever is happening right now on the screen. But believe me, I was a student who was so happy during my 12th standard result just because I passed in maths. I got fantastic marks in other subjects, but I was so happy I passed in math because I didn't want to do anything with maths. But when you come to BSc, you realize math is an integral part of your entire uh, understanding of biology. It's just not mapping of genome what is taught in your syllabus, but the any research project that would you would take up, and you need to do biostatistic, you need to know what is the standard deviation, you want to know whether 100 patients given 100 treatments, and what is that which will give you the correct treatment with the correct patient. So everything finally boils down to math. And do not get worried, because all these are computer driven software tools which will make that math happen for you. Okay, so do not get worried. But as a teacher, I think what I tell my students, I will tell you all as well. Try to do smaller courses in biostatistics if it is not there already in your syllabus. Or there are some very beautiful courses on Coursera, very interesting ones, very knowledge building ones. So do look at them. You cannot, as again and again I keep telling, compartmentalize yourself into one discipline. When we talk biology, when we talk bioinformatics, learn Python, learn basic C++, because there is Java involved in small programming. And let me tell you, some of my students who went to US or some other labs, they had to do the programming part themselves, because nobody is going to do this small programming for you where you are interested in one small genome of some bacteria. Nobody is going to give you that, and the earlier which is available kind of a algorithm is not working for you. So what you need to do is you need to have these capacity building attributes collected over these 3 years of your BSc. I think it's enough time for you to become an all-rounded personality who can then pick up the tabs when you go for your MSc, those who are interested in doing bioinformatics. And the best part of bioinformatics, again, I would tell you is that, what is this lockdown time, right? We are at home. And I would learn a lot of bioinformatics just sitting at home with a click of my mouse. I do not have to go anywhere. All my bioinformatics softwares, all my tools come with inbuilt query solving machinery. So I really don't have to go out. I can sit here. Lockdown doesn't deter me from learning bioinformatics. So imagine that. And bioinformatics is also for those people who don't like to be with too many people, chow, 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 crowd, ye, wo. You don't have to be. I'm not saying you have to be isolated, because again, you have to share your data. You have to share your outcomes with other scientific community to validate what you're doing is right, to learn whatever you're doing is correct. But you can be by yourself and learn a lot of things if you are a bioinformatics learner. So as you can see here, there are a lot of new things that you need to learn when you get into bioinformatics. I cannot really go ahead unless I talk about few of these people. And I'm sure some of you, see, those who do not respect history, I think they cannot build a good future, So that is why when we talk about bioinformatics, don't learn it as a chronology thing, learn it as a story. And therefore, Margaret Dayhoff, I call her mother of bioinformatics. Remember, if you go to her biography, she was such a wonderful woman. She was a chemist, mathematician. That time computer, nobody heard the word computer, okay? And I'm sure you must have got the pictures of those big sized, oversized, like a big cupboard kind of a computers, that time. So she did all that, and she came out in 1965 a protein atlas with protein structures. Imagine the cleverness or the insights that she collected, being a mathematician, being a chemist, and being a earlier first, I would say, woman bioinformatician. That time the bioinformatician word was also not there. I'm sure you know Frederick Sanger, DNA sequencing, because everything small dots you need to connect when you're doing bioinformatics. Eugene Mayer, I'm sure everybody knows NCBI. And before going to Eugene Mayer, I would say David Lippman. He was a director at NCBI. And remember, an institution never grows in multiples unless they have a very good leader. And David Lippman provided that leadership strong. So therefore, NCBI is now the pivotal agency which can help everyone with the bioinformatics, any kind of information. Of course, Eugene Mayer, known as Gene Mayer, he is an American computer scientist and bioinformatician, and he's best known for his BLAST algorithm. Craig Venter, I'm sure those who are doing biotechnology, they know Craig Venter. Craig Venter was one of the scientists involved in Human Genome Project. Later on, he had big fallouts with the team of Human Genome Project. He started his own company, and he's famous for Cynthia. Cynthia is the first artificial microorganism with a artificial DNA that was created. That is Craig Venter for you. And of course, I could not resist Vasant Honawar. He's an Indian-born American computer scientist, very well versed in artificial intelligence and machine learning. So all these people, I think they have built this field of bioinformatics. And when I did bioinformatics, when I did learning, I learned a few things in bioinformatics. That was 30 years ago. Now things are changing so fast. Every day there is a new software. Every day there is some new breakthrough. So you need to be really updated about whatever has been happening before and how fast it is changing. Of course, you are free to, learn about these stalwarts as well, because we always talk about history. We always talk about people who are no more, and we try to glorify what they have done. Of course, they are great. But remember, why I have put this slide here is because those who are interested, now please be, I'm sure your teachers have told you this also many times. Be vocal, be connective, be always on the lookout where, who, what is being doing in what labs. So tomorrow, if you want to join their lab for an internship, you can directly write an email to them and say, sir, I have looked at your profile, I have looked what happens in your lab, and I think I am interested in joining your lab. This networking will only help you. Knowing only about history and not knowing about the current scenario or the current work profile that is happening. And again, another thing I want to tell you, our labs, Indian labs, are as good as any foreign lab in today's time. We have the same intelligence pool, we have the same technology, we have everything similar. So do not have this golden nest kind of a thing in your mind कि अरे मुझे गोल्डन पंख तभी लगेंगे जब मैं यूके या यूएसए जाके पढ़ूंगी या पढ़ूंगा. Please do not have that kind of notion anymore. In fact, India is going to be one of the superpowers, and this is not just khayali pulao, because these are the people who are making the new India happen, and these are the bioinformaticians who are going to build further roadmap for bioinformatics. So now, what are we doing? There are some lighter moments here. The cartoon says that the good news is that we have the human genome decoded, but the bad news is that the computer has alphabetized it. So imagine what must have happened if this happens, really. If all A's come together, all C's come together, all G come together, imagine. And of course, the God is not really happy that the human beings are knowing about their secret code of genome. And then he says, damn the hackers, now I have to change the password. So after human genome, what did happen? There was an aftermath of sequencing. That means they definitely wanted to know many things, but those understanding about genes, coding, non-coding sequences, open reading frames, where a gene starts and where it ends, all that actually led to a lot of other things. And that is why we needed to keep a record of all normal and variations and polymorphisms. We needed normal to abnormal records. We needed to know the mutations. Just imagine 3.2 billion base pairs that we have in our genome. Imagine the mutations, imagine the back mutations, forward mutations, and imagine the total rate at which our genome is changing. Even if we want to keep record manually, I don't think any one of us is capable of keeping our own record of our genome, let alone the entire world. So definitely we need something like bioinformatics. And yes, these are all the species and coming in more, those who have been completely decoded and the information is being organized. So therefore, why and where do we need these databases? We need these databases to keep that all information. And today you can click on any of the database and retrieve any information that you need. So you have sequence databases, you have structural databases, you have genome databases, and you have proteome databases. And I'm sure all your teachers are going to teach you this, or they are going to tell you some wonderful things about these databases. So if I have a species database or genome database, I can just go on that. And if I want to know about Drosophila, I go to Fly database, Flybase, and I will come to know everything about Drosophila, right from developmental genes to all the genes in that. So basically, databases help us in collection and organization of the data. A little bit here, I'm sure you have seen this, all the TYs, SYs, those who have already gone through this. But what I feel and what I really insist is, just don't go here and follow what your teachers tell you. Okay, click on all databases, select nucleotides, select protein sequences, put your name of gene here and just go ahead. No, please. Once this practical is completed, go on all these popular resources here. Go on all these resources on this side, because we don't get time during the regular practical. We kind of take it for granted that, oh, students are already knowing about all this. And please don't do that. I'm really urging you. This is all self-learning thing. Please go ahead. So the second page that we see is something like this. And often what we do is, whatever is this interested genome that we go click on it and we try to see the next page. What about all these filters that are there on your left-hand side? What about these? You can actually search molecular types. You can search, all RefSeq sequences. You can search lot of things which are customized. Please explore each of the tool that is given to you. And finally, of course, we go to the gene bank main page. As ma'am said, yes, I have some sequences here. Thanks to my PhD student, Sharon, we were working on plant, chloroplast, and mitochondrial genome. And yes, we did find some very interesting ecotypical genome variations. But coming back to what you are seeing on this, imagine you are looking at this as gene, then exon, CDS. How many of you really know the difference between exon, difference between gene, and difference between CDS? We just go blindly following. So when I'm training students in databases, or whenever I tell my students to learn about a database, my very focus is just for one practical, I'm going to show you this one page, and then let's each try and understand each and everything that is written here. Let's not go ahead in a hurry. But finally, of course, we are going to see this sequence and all these ATGC base pairs. So that's fine. But before that, I want to know what this exon means, what these numbers means, and then only I will be satisfied. So ask questions. Keep asking questions to your teachers. If they don't know, they will find out. You must have seen PDB. I find this a very nice protein database or protein data bank, because your COVID-19, amazing and immense information regarding COVID. So even if you are just wanting to explore PDB, go here, type COVID, and you will be flooded with information. So coming to the tools used in bioinformatics, there are so many tools. As I said, there are many tools in bioinformatics. You don't know which one to use. So basically, you have to first go with your teacher. In fact, ask her, what is the task that I am assigned? Am I assigned a task of comparing two sequences? Then I'm going to use similarity tools, and that is my BLAST, basic local alignment software tool. That is what I'm going to use, because I'm going to compare two sequences. What is the next thing? Am I doing any phylogenetic analysis? Am I looking at two sequences, one from mice, one from human beings, and am I trying to search for my ancestor? Then I do sequence analysis. Then I look for phylogenetic analysis. I look at common ancestor, and then I say, oh, I go back to that. This gene goes back to bacteria. Am I trying to look at a novel protein that I have, and I don't know whether this protein is new and I can name it after me, or is this protein same as it was somewhere I can find it, a similar sequence of protein? Then I do structural analysis. Then I look at some predictive tools where protein structures are there, some scaffolds are there, where I can put, like, it's scaffolding is like mannequin, you have a mannequin in the shop and you put clothes over it. Something similar, you have a polypeptide sequence, okay, and you get it into primary, secondary, tertiary structure, and finally there is a scaffold of protein original, okay, in the database. On it, you can put your new protein and see how well it fits. So that is your structural analysis. And if you can do that, but for that, first you must know what a protein looks like. What is the difference between primary, secondary, and tertiary structure? And the BLAST, FASTA, emboss is a molecular biology tool by European EMBL. Clustol W, RASMOL is something where you can predict structures, and I'm sure your teacher will show you sometime the application of RASMOL, which is very interesting. And you have application programs like BioJava, BioPerl, BioXML. You can get into it a little bit, but I think first you need to make your basics straight and comfortable. Then I come to application, and these are some of the applications are new. Some of the applications are there from quite a few years that they have been going on, but they are being labeled differently now. So where are these applications of bioinformatics? And if you see this, even a botanist can become a bioinformatician. Even a zoologist can become a bioinformatician. A microbiologist can become a bioinformatician. A biostatistician also can become a bioinformatician. If you are learning clinical, research, you can become a bioinformatician. So just look at this list. I don't want to get through the entire. An environmentalist can become a bioinformatician, because finally you are looking at the DNA, RNA, protein of a cell and how it interacts with its environment, or how does it adapt or change to form many variables which can be used for human life, finally, of course. So some of them, I'm sure, structural genomics, you know how DNA sequencing and Sanger's that I just mentioned, Sanger's Institute is one major work has been done in sequencing. Functional genomics, as again, when I talked about stimulus, response, and the gene expression, I always like to give this example to my students that yes, I have a pretty plant with very bright red flowers, as long as I keep it outside in the garden. The minute I get that plant inside my home, the flowers try to change their color to pink or sometimes colorless. And then I wonder, why is it so? And that is all because the stimulus and the response that is given by the gene, as to if it has bright sunlight, it will be brightly red. And if it does not have that enough sunlight, there are some enzymes are not going to be working there, and you're going to have a pink color flower. So that's the most simple example. So when you start with bioinformatics, you start at the genetic level. Then, of course, you look at the proteins, and then overall how these proteins interact in the cell, and finally at the organismic level. So no information is, not required, I would say. And comparative genomics, I'm so very, every time I see this picture, something else comes to my mind. And if you can see these horizontal lines here, if you can see these horizontal lines, okay, you can see all these letters. And because I'm not taking a class with you, I'm giving you an overall overview of bioinformatics. I'm just telling you how you can compare the genomes. And these are single letter code of amino acids. And this is a particular protein or a particular fragment of one individual, second, third, fourth, fifth, sixth, seventh, eighth, ninth, and 10th. And they are just being juxtaposedly placed next to each other. And they're going to look at similarities and differences. That's your comparative genomics. And comparative proteomics, because these are the protein amino acids that they are written here. And can you see this? All these WWW here and one F here. So here is a mutation that has happened. Okay. And here, this is our forefather, of course, and he's telling the dog that, hey, we are related. And I'm sure the dog is not very happy. And he says, really? And I think why the dog is not happy, because he says, I have a loyalty gene much more strong and much more robust than you. the loyalty gene in humans have got mutated. So I don't think I'm really going to be happy about being related to you. So of course that's just in a lighter note, but yes, you can actually compare thousands of sequences, and that's exactly what BLAST software does for you. It compares thousands and thousands of sequences in the database to give you all those which are similar and filter away those which are not similar. Proteomics, this is the hot field nowadays because genomics is really worked upon a lot. Reverse genomics is something people are looking at, but yes, proteomics is there. And there you look at the protein structures, you look at their expressions, you look at protein-protein interactions. And this is where the gray area is, because at any given point of time in our cell, I think we have around, please correct me if I'm wrong, around 50,000 proteins, small, big. And if they all are interacting with each other in that small space of cytoplasm or nucleus, you can imagine if they are not really on good terms with each other, what is going to happen. And therefore, proteomics poses a bigger challenge to us as to how are these proteins interacting with each other, and how does it then bring a different kind of reaction or expression in the entire profiling of a system. Structure-based drug designing, this is the hottest field, and everybody, whenever I want to give people kind of a positive picture, I say, oh, you need to do drug designing, you need to get into this field because you'll be designing a drug. This is how it is done. Of course, this is one disease gene, they have found out here on a chromosome. Then you translate the DNA sequence into amino acids using a software tool, of course. Then you find similar sequences in the database, and then you use one of the animal model, uh, which has a similar protein, of course, and that protein then will be reacting with drugs. And then once it is in silico, done in silico, it can be done in that worm or in that microorganism, and whatever responses you get, then you can scale it up, then you can extrapolate it to other systems of fly or human beings. So imagine this whole journey is like not very long, just 10 years or something like that, to find a new drug. So how much patient are you in this whole process? But definitely, if you do in vitro, whatever takes 1 month, it is done in 1 day, or sometimes even less than a day when you do drug discovery. Because what you're doing actually is you are actually looking at most close molecules which will give you a positive result, and you can negate rest of the molecules which otherwise you will have to do these lab experiments for every molecule. Here, by in vitro analysis, you can, out of 100, you can choose 2 molecules and go ahead. So your time, money, energy, everything gets saved. So this is molecular docking, another interesting field in bioinformatics. And docking is nothing but the target molecule and a ligand. I'll give you a very simple example before we read whatever is there on the slide. Is that if you are coming out with a new drug, okay, that drug molecule, how effectively it fits into the target molecule to bring about the desired effect of, say, pain blocking. Now, this example here I have quoted is HIV-1 integrase, a new binding site for drug treating AIDS, was discovered by Shams et al. Using docking studies while considering the flexibility of a receptor through molecular dynamics. So there are many, again, software tools. Autodock was one, in conjunction with relaxed complex method to discover novel mode of inhibition of HIV integrase. It is an enzyme of HIV, which if you inhibit that, you can stop the spread, or you can check the spread. So this is so easily said, right? As I said, when you do molecular docking, you need a lot of other basic knowledge that needs to be with you. And yes, as I'm a botanist, I always am true to my alma mater, and molecular docking can help in so many things in food industry. There are huge number of articles on the web for that. And I have included one very interesting example for all of you to see, okay, or to learn today. Yes, this was again the computer-aided drug design that I was talking about. So CAD is a special. This is a relatively new one. CAD is a specialized discipline that uses computational methods to stimulate drug receptor interaction, and everything is, of course, bioinformatically driven. Also, there is a media hype. Remember, people think, oh, if I know a little bit about genes, if I know a little bit about some software tools, then I can become a bioinformatician or I can learn bioinformatics. Or the second and very, very dangerous grounds are bioinformatics can do anything. And I'm going to really, I'm going to tell you the finer difference between the in vitro and in vivo kind of or in silico kind of a experimentation. Uh, when you do anything in silico, I wouldn't say there are no quality checks, but there are fewer quality checks. And I will tell you why I'm saying this. Many people have very limited knowledge of the subject, and if this knowledge is available online free, then you can imagine there are more users than developers. Okay, so everybody and anybody is going to use this, and therefore you have an element of that is confusion. And of course, people are always confused when they are working in bioinformatics. And that is why I said, if you are really, really interested, get connected with a lab which is working. Nowadays, they allow people, students to get involved if the student shows the promise. So, a note of caution. False positive or false negative need proper interpretation. Computational tools are predictive and not confirmatory. So here you see this picture and people must be wondering, why is this very nice picture here out of context? Okay. And here, can you see this picture here? There are two receptors and you have to choose between which one is there, which is going to really make sense and give you a better medical output or a drug output. And here everything, oh, he has a laptop, he has a desktop, he has some coffee tea thing, he has a mobile, and he's happy. He says, oh, I don't need to look at anything else. Now I go back to this story, and suppose there is this boy who wants to propose to a girl. Okay, and the chances of that girl saying yes to him can be 50-50%. I hope everybody agrees with me when I say this, and I will tell you why I'm taking this analogy. Okay, this boy says that, I want to propose to this girl, and his friend says, so what is the possibility of that girl saying yes? It's 50-50. And that boy says, okay, if it is 50-50, then I want to make it 100%. And how do I make it 100%? By changing parameters. And that is exactly what you do here in silico when you're working. If you're giving, if your target A is giving you a certain output and you want more output or more close docking between the drug and target molecule, I'm going to choose another receptor. So that means I'm going to change the parameter. I'm going to bring in more variations. Then I may change the pH also. Then I may change the temperature also. I may try to manipulate my external conditions. So something similar here. The boy says, oh, I think the girl, somebody told me that the girl likes a boy dressed well dressed. So whenever I go to propose to her, I'm going to dress well. So this is one parameter that I'm going to bring in. Next, he says that, they tell me that she is very in a good mood when she goes out with her friends in the evening. So the second parameter comes in, and the boy says, okay, so the timing that I'm going to choose is in the evening when I go and propose to her. So he has already manipulated the environment so that his output or the answer that he wants to get, like how here we want to get the most effective drug, needs to be 100%. Okay. And if I may ask, so in spite of doing all this, when he actually goes, meets the girl and proposes, what do you think would be the answer? What do you think? Let me tell you, there is never 100% guarantee that the answer will be yes, and all girls will agree with me here, isn't it? And all boys, of course. But then that exactly what happens when you work in silico. When you work computer assisted drug designing, when you work on all this, all you're doing is you are narrowing your search. Never think for a minute that if I don't do wet lab experiment, then also my things are going to be all right. Never. So there also always has to be a backup for R&D. Whenever you're working, there has to be a wet lab experiment confirmation whenever you are trying out a target molecule or some lead molecules which need to be tested. And those testings first, of course, whatever layer of clinical testing it has to go through, and then finally the drug comes out in the market. So there is no exclusivity, no mutual interconversion of in silico and wet lab experiments. So you as biologist, again, I come back to the same thing. You have an upper hand because you are a biologist. You can do wet lab experiments. So what's the scope? Everybody would ask me, then if I become a bioinformatician, where would I be employed? And you can be a biostatistician, you can be a pharmacogeneticist, you can be a bioinformatics software development. If you are good at computer and if you learn a little bit of programming, you can be that as well. Of course, you can be a genetic analyst, and you can also be scientific curators. And there are really many opportunities. The pay is good. Indian scenario also, many companies are now looking at big data. They are looking at machine learning, and they need bioinformaticians with biology background. So if I'm a bioinformatician, quickly before going ahead with the last slides of mine, which I'm going to talk about, a specific example as it excited me actually. So what if I'm a bioinformatician, what will I do? So I may envision and build a database for researchers to share their biological data and observe how they interconnect these data. Or I could help to create a personalized medicine by working with a team to evaluate thousands of individual genetic code and the biochemistry. So I'm going to look at their SNPs, and that is single nucleotide polymorphism. Or I can create a computer tool to track and analyze the patterns of viral outbreaks like flu, COVID-19 around the country, right? Or I can be a programmer or program an algorithm that will help piece together the structure and function of a protein. So sometimes the structure is known, function is not known. Sometimes function is known, structure is not known. So I can be that as well. So if this excites you, bioinformatics is the field for you. So here comes an example which I was intrigued by this. Have you heard about Impossible Burger version 2, 2.0, as it is said in today's language, and the next generation of food tech? Now, Impossible Foods is a company which is established in, I think, 2015. And imagine this, it is doubling its R&D team and seeking world's best scientists to help eliminate animal agriculture. This is exactly how bioinformatics is translated into a successful business model. So those who are entrepreneurs here, please pay close attention to this success story of Impossible Foods. The name of the company is Impossible Foods. So what did it do? It developed plant-based substitutes. So researchers, what they did? Animal products at the molecular level, they researched it, then select proteins and nutrients from plants to recreate the experience and nutrition of the meat product. So they are completely trying to replace the meat. And that is exactly what the burger was shown about. A Stanford biochemistry professor, Patrick O'Brown, spent an 18-month sabbatical. He went on a leave. So it's a good news for all our teachers who want to do something similar. So he went on investigating how to eliminate intensive animal farming, which he determined at the time to be the world's largest environmental problem. Really, because when you want to rare animal breeding karna hai aapko, then you have to depend a lot on their food and their nutrition. So finally, ultimately, who's getting affected? The plant, plant world. So the company claims that making it uses 95% less land, 74% less water, and it emits about 87% less greenhouse gas than making a ground beef burger patty that comes from a cow. Imagine this. I mean, what a novel idea. The plant-based burger has more protein, less total fat, no cholesterol, and less food energy than a similar sized burger patty made with beef. It contains more sodium and more saturated fats than the unseasoned beef patty. On January 7, 2019, it is this time that I'm talking about, Impossible Foods launched a new version for their signature burger, the Impossible Burger 2.0. The company has stated that the new burger is tastier, juicier, and more nutritious, featuring 30% less sodium and 40% less saturated fat than our current recipe, which was the initial one, and just as much protein as against 80-20 ground beef from the cows. CNBC, one of the leading news agency, reported that coronavirus pandemic generated new interest in meat substitutes. Everybody, you know vegan people, there are many people who are going off meat nowadays. So people are definitely looking at plant-based substitutes which can be alternatively used. So as COVID-19 outbreak in meat packing facilities caused shortages, they were looking at other alternatives. And in February 2021, that is just recent, what did company do? They were very smart. They announced that it would be lowering US grocery store price of its product by 20%, which actually increased their profit. So what was this all about? As a bioinformatician or as a biologist, all this company talk is good, but what exactly was happening inside that burger? So scientists at Impossible Food found that heme is the key factor in how the meat behaves. Heme is the molecule that gives blood its red color and helps to carry the oxygen in living organisms. We all know that. Plants, particularly nitrogen fixing plants and legumes, also contain heme, which is called as leg hemoglobin. And if it is very interesting to search the phylogenetic lineage for hemoglobin molecule, it's very, very interesting. So plant-based heme molecule is identical to the heme molecule found in meat. And how did they come to know about this? Using bioinformatics, using the in silico model of protein structures. Then they came to know that heme molecule is identical to the heme molecule of meat, what is found in plant. Then of course they selected the hemoglobin molecule found naturally in the roots of soy plants. To make it in large quantities, they genetically engineered it using a yeast and used a fermentation process to scale it up. So I'm sure here those who are microbiologists or those who are biotechnologists, they would be very happy to read this. To replicate the fat in hamburger made from cows, Impossible Food used flakes of semi-dried coconut oil, which were mixed with the ground textured wheat and potato proteins. The potato protein provide a firm exterior when the meat was seared, and the coconut oil stayed solid until heated, when it melted in a similar manner to the beef fat. So what they were trying to do, they were trying to actually make it taste just the same so that people would go off meat burger and would come in for this. So money matters. Impossible Food has raised round of $75 million and 108 millions from investors, including Google Ventures, Khosla Ventures, and even Bill Gates. And it was reported that Patrick O'Brown, the CEO and the founder member, had turned down an offer of $300 million to buy out Impossible Foods in 2015. Imagine at that time it was to be bought at this cost or price. Imagine it today. This is another company, Beyond Meat, which is also using bioinformatics to make pizzas, which are vegetarian sausages specifically, but which are vegetarian sausages. So I'm sure all those who are listening to this talk today have got a lot of bright ideas in their mind, and they want to do all this, and they are rearing to go. People may ask me what is 3D bioprinting and bioinformatics in common. And I'm sure some of you may know what is 3D bioprinting, because it is used to create tissues and organs where biological cells or biomolecules materials are used as ink to fabricate 3D biological structures. And where do these 3D biological structures come from? They come from the basic research, basic findings from your DNA, RNA, and proteins. And that is how bioinformatics is in a way connected to this 3D bioprinting, which is the next gen thing that all of us will be witnessing. Scalable, efficient, and economic manufacturing solutions for key raw materials such as recombinant protein bioinks, growth factors, and other maturogens will be essential for successful commercialization. They have not yet mastered this technique, but a lot of research, a lot of funding is available in this field. Creating training databases for big data curation, building digital twin of human organs with cellular resolutions and properties are some of the most important and urgent challenges that this field is facing. I'm almost at the end, and you can tell me to stop anytime, ma'am. So since I had said I will be talking for an hour, but It's my
Ma'am,
passion.
you can continue for as much as time as you want. No worries.
No, no, no.
I
This
believe
is
all
just
the participants are definitely enjoying it.
thank you so much. No, just few more slides because people would want to know. We talk about all these big things. Okay, we talk about 3D printing and we talk about replacing all the meat products by plant-based products. Finally, we have to start somewhere at the beginning. We have to start at BSc level. We have to start at the beginning, right? So please do take interest in whatever your teachers teach you during your undergraduate years. And these are some of the colleges in Mumbai, I would say, that would offer you bioinformatics. And then, of course, there are a lot of institutions across India, abroad, who will also offer you a lot of bioinformatics training. As I mentioned, I think before, Coursera is another portal where you can find some very basic bioinformatics courses. But I have found some students who go there without basic preliminary studies or preliminary information. They find Coursera difficult. So you have to choose a course accordingly, and you also have to build up on your database at the preliminary level to be able to handle the courses that you want to take online. And as I said, your teachers are definitely there to help you. Some of the entrance exams, these all exams have lot of bioinformatics questions in that. And NET has bioinformatics in that. PET has bioinformatics. There is some bioinformatics national certificate examination also there, which is based on bioinformatics. All these GATE, BARC exam, TIFR, ATREC exam, all will talk about bioinformatics in some or the other form. These are some of the resources, very basic books, but as we are under lockdown, we can definitely have these e-copies of these books. And I remember when I started doing bioinformatics many years ago, Dr. Medha Rajadhyaksha, who was that time the HOD of Life Science Department, Sophia College, she gave me the first book which I actually used, and it was called as Bioinformatics for Dummies. And I was literally a dummy that time when I wanted to learn everything from basics. But nowadays you have a lot of self-learning tools. YouTube are filled with bioinformatics videos. Make the best use of it, I would say. Another thing I would say again is, though I'm a plant scientist, or I will not call myself a scientist, but a student of plant science, still being in life sciences has given me this opportunity to go beyond my things that I have learned in college, in my undergraduate, postgraduate, and PhD. So I could expand my horizons, learn about animal systems. I could learn about bioinformatics more, because when we talk about bioinformatics and we talk about drug development, basically we talk about microorganisms, and then we talk about animal models, because plant models, few are there, but the response or the mutations or the expressions of the genes to detect in animals is faster in those models than in plants. So I did come across one very good report where there were stress tolerant genes detected across a huge genome of plants using bioinformatics. So if anybody is interested, any of the teachers, I can share that uh, PDF with them as well, because a lot of insight on how that also can be given. These are my references. All this information is available on the net, as I said. And I think, yeah, that's it. I think that's all from my side. If anybody wants to know anything more, you are free to ask me questions.
Ma'am, that was a wonderful talk. Just one question we have, so I can declare the question to you. There's a person who's asking, among plant genomic databases, which database should be preferred?
So we have always gone back to Arabidopsis, and I was fortunate to visit Max Planck, because of one of my student was doing her postdoc at Max Planck, and their bioinformatics center is fully equipped with plant genome database, which is basically dealing with Arabidopsis genome.
Okay.
So yeah, or nowadays they look at maize genome, but more results or more quick. I would say, like I said, model organism, right? It's Arabidopsis. So many genes are tested in that. Yes, that's what I would say.
I think there is also plant GDB, which is most commonly
Ye-
used nowadays, which has a
Absolutely,
lot of
absolutely.
articles written about it.
Yes,
Plus, you have
yes, yes,
specific
yes.
ones on Brassica and things like that.
Yeah, yes,
Yeah,
yes. So again, as I said, you can go across
yeah,
the net, and there are lots of things. Newly, there are many things that are being introduced, because something what is understood before may not hold valid now. So there are very many new researchers. Yes, they are coming up, and so one activity we do at our end is when we do our SYBAC and TYBAC practicals. At each practical end of it, we give a topic to the student related to that practical, and we tell them to find out research papers based on that. So what happens is, and we have made groups of students, so each group comes out with one paper, and they
right,
read the whole paper and they try to understand what is exactly given there. So that has also added to our pool of resources,
right,
because
right.
me at my end I can see only one paper. If I have five groups, I get in five papers, so it's like that.
All right. Yeah,
Yeah.
that's a good
Yes.
way of doing it.
Yes.
Yeah,
So
Vivek, your question is answers. Rodhan Patke says, thank you so much, ma'am, for such an insightful session in the avenue of bioinformatics. I think, yeah, it was certainly an overview, but it was one of the most, I think you went into giving the crux of what was supposed to be done out of that whole thing. So I think the students certainly are inspired, and I hope that my students pay more attention now in class post this lecture, because of the fact that, like we just discussed, that virtual learning is very, very difficult. Bharti ma'am, are you there? Virtual learning makes it impossible. So I thought that I'll ask them to do it on another phone and show it to me as a part. So I hope they will do that.
yes, yes. I
Sagarika
actually,
madam,
yes.
Sagarika
Sorry.
madam, thank you very much with respect to the kind of talk that you have given an overview of bioinformatics, which has always been a challenge to take or teach, with the kind of knowledge that we all have presently and the way that it needs to be told to the students. They should know everything about a little bit about genomics in detail, as you said rightly, transcription, post-transcriptional modifications, splicing, etc. Then whether you talk about the cDNA sequences or the messenger RNA sequencing and all, until they understand the basic genome, it's a little difficult for them to
Yeah.
understand this particular branch of science. But the way in which you have put in that what all it gives you finally when you do it this way correctly, that was a wonderful way in which you have really put in front of these students. And when we are going to take bioinformatics for TYs in this term, I'm sure now they are going to enjoy better, and they are going to ask us more questions also based on what you have asked or you have told them. Very, very,
Thank you.
very beautiful lecture, very beautiful presentation that you have made, I must say.
Thank you, Sangeeta ma'am. I think we all have become equipped in making presentations, if nothing else, during this pandemic, right? I mean, as I said, I'm technologically challenged person. I too can learn. So our students are great, and I think all you leaders, teachers who are there, I think it's going to be a win-win. And please do encourage the students to get into bioinformatics, because I will tell you very honestly, I have every year I have at least 4 to 5 students opting for bioinformatics MSc, and then they enter to the field of research or job. They still are using that information somewhere, someplace. So no information ever goes waste. And if
Definitely, we will do that. We will do that.
yeah, and please try to get in little bit of Python or little bit of C++, because we don't have to write algorithms, but we definitely need to let students know that they might have to encounter it some or the other time. So
Sure, we will do that. That's
yes.
a question. We will definitely look into that.
Absolutely, yeah. And sorry, I could not show you any magical things about rotating protein structures and those ball and stick models and all
That's
that.
okay. That we do when they're doing
I
as well as practical. But to
exactly.
understand
So
what they're doing is more important. Yeah, yeah.
absolutely,
That was
absolutely.
definitely required. What you told, the different branches, proteomics, genomics, and how they're merging everything. and docking and all. These are the things that they should know that is encountered in bioinformatics. That was nice.
Thank you. Thank you.
Yeah, back to you, Archana. Sorry for interrupting.
No, no, ma'am, not at all. Bharti ma'am, it's over to you.
Yeah. Good evening, everyone. On behalf of Botany Department and Jain College, I would like to express a very hearty vote of thanks to Sagarika ma'am. Thank you, ma'am, for giving or taking this opportunity to actually give us immense knowledge and sharing your valuable opinions on this topic, that is bioinformatics. Thank you for your precious time. We have thoroughly enjoyed your talk, ma'am. Thank you for giving us taking this opportunity and giving us a very informative talk today. So, also, along with this, I would like to thank our principal, Dr. Ashok Wadia, for always encouraging us to take initiatives for both teachers and students alike. And at the end, I would also like to thank all the teachers and students who have participated in our lecture series. Thank you everyone for being there for this lecture. So,
Thank you so
thank
much.
you so much, ma'am.
Thank
Thank you,
you
ma'am.
all. Thank
Thank
you,
you.
Devangi ma'am, for inviting me.
Thank you so much, ma'am. It was a pleasure. It was really wonderful to hear you.
Thank
It was an absolute different way in which I believe now students will look at bioinformatics,
I hope
which
so.
was always considered one of the most adult topic, which would just talk about most basic, tools and etc. etc. Now they will at least look at it in a different perspective. So
True.
thank you so much for changing it, ma'am.
Thank you so much. Thanks. My pleasure.
Thanks, ma'am.
Yeah. Can we?
Yes, ma'am.
Yeah.
Devani ma'am,
Thank
you
you.
end the broadcast.
Yes, ma'am.
Thank you,
Thank you so
everyone.
much, ma'am.
And
Thank you,
thank you
ma'am.
all the students. There were no questions, but I hope that means they have either understood or not understood. I don't know. I'm an
Majority of them have. They have at least got some
thank you.
inspiration. That's what the WhatsApp
Thank you.
messages say. So I
Thank
hope
you.
they are inspired.
All the best. And anytime you need any help from my side, I'm always there.
Certainly, ma'am.
So, yeah.
Will
Thank you. Yes.
I will
Thank
be
you, ma'am.
contacting
Thank
you.
you.
Yeah.
Bye.
Thank you.
Take care. Stay
Bye.
safe.
Thank you so much,
Bye,
Stay
ma'am.
ma'am.
safe.
Stay
Thank you.
safe, ma'am.
Yeah.
Yeah. But Devani ma'am, your broadcast needs to end.
I'm just sharing the